Hello guys, welcome back. This is Dr. Hadi here. Today's topic is cofactor and coenzyme. We have studied a lot of videos about the enzyme in this chapter. This will be another interesting video. In today's lecture, we will discuss what are cofactors or what are coenzyme. What is the role of coenzyme or what is the role of cofactor? So here we start with the definition of cofactor or coenzyme. Here it is mentioned the non-protein part of the enzyme which is necessary for the proper function of enzyme. Here in the diagram you can see this diagram is of an enzyme, and we know very well that this enzyme is composed of smaller units called amino acids, right? So I can write here amino acids, large number of amino acids are assembled to form one complete enzyme. It means we can say that amino acid assemble to form what? To form enzyme. And we also know that the combination of amino acid is called protein. So we say enzyme is made of what? Protein. This structure which represent an enzyme is actually a proteinous structure. This whole structure is protein. Okay, in majority of the cases, enzyme itself can catalyze a chemical reaction very easily. But in other cases, the enzyme alone cannot perform its function. Why? Because such kind of enzyme, they require another friend, they require another factor, which is called as cofactor. Co means friend, cofactor. So here I have mentioned another thing that is called as cofactor. This enzyme is waiting for the cofactor. Once this cofactor attaches with the enzyme, then this enzyme will become complete enzyme and then it can start its function properly. So until and unless the cofactor is not attached, you can only call it just an apoenzyme. This is just what? This is just an apoenzyme. I can write here apoenzyme. So this is our, in the beginning of the video, I called this structure an enzyme. Now, little bit advanced, I call him apoenzyme. Why? Because its friend is missing. Its friend is cofactor. So I am going to bring this cofactor and attach it with the enzyme. So here is our cofactor. This is now attached. The combination of cofactor and apoenzyme plus cofactor. Now these two will combine another third thing that is called complete enzyme. That is called what? Complete enzyme. Complete enzyme. Because this enzyme is now capable of catalyzing a chemical reaction. The complete enzyme is also called as holoenzyme. It is also called what? Holoenzyme. So by definition, you can write the definition of holoenzyme is an apoenzyme with its cofactor attached is called holoenzyme. Here you can see apoenzyme plus cofactor is equal to holoenzyme. So today's lecture is about this cofactor. This cofactor. Here I have mentioned. Now you got the idea what is cofactor. It is non-protein part. Okay, This is the protein part, the red one. The red one is, of course, amino acid. The red one, this is the protein part. Protein part. And this one, this part, the cofactor. This is what? This is the non-protein part. This is non-protein part. In the book of Satyanarayan, most of the cofactors are non-protein, but I saw one cofactor, one coenzyme, which is protein. I will also discuss this with you people as well. But in majority of the cases, this cofactor is a non-protein part. Okay, so cofactor here, there are two arrows. Cofactor, this one may be organic or inorganic. In case it is organic, it may be loosely attached or strongly attached. If this cofactor is inorganic and attaches loosely, again just write a dotted line, loosely attached. If it is if the attachment is such like that it can be separatable, then this will be called what? Coenzyme. Now the students get confused between these two terms, the coenzyme and the cofactor. Cofactor is a general term that can be anything, but coenzyme a specific factor that can be special thing if it is organic and loosely attached. So a cofactor become coenzyme when it satisfies these two conditions. If a cofactor is organic and if that cofactor is loosely attached, then the cofactor will be called coenzyme. The biochemist usually use the word coenzyme for all of these cofactors. Okay, so there is no problem. If you use this word interchangeable, you can say cofactor or coenzyme. But being a student, you have to make difference clear. Okay, if the cofactor is strongly attached, now I am going to write a shaded line, means that the cofactor is strongly attached with the enzyme. It cannot be separated. So in this case, it will be called as prosthetic group. So the comparison between the cofactor and prosthetic will be that a cofactor becomes prosthetic group if it is organic and it is strongly attached. So a cofactor must satisfy two conditions in order to become a prosthetic group. Then if this cofactor is inorganic, if it is inorganic, maybe it is an ion, calcium ion, magnesium ion, chloride ion, zinc ion, manganese ion. In this case, these cofactors will be called activators. So now we have three types of cofactors: activators, prosthetic groups, and coenzyme. These three are cofactors. But the biochemist can use the word coenzyme interchangeably in place of prosthetic group and activator. Sometimes if you see in reality it is a prosthetic group, we can still call it coenzyme. It's okay, fine, but difference is necessary still. Okay, the idea is clear. Now I would like to tell you what is the role of coenzyme and what is the characteristic of coenzyme. Some points are being mentioned on the whiteboard. Each point will be discussed. Point number 1. The coenzymes are involved in transfer of atoms or groups. Means, let's suppose I'm going to erase this diagram, or I can. Let's suppose this is one enzyme. This is our enzyme. Okay, this is our enzyme. I can write here E, and the cofactor is attached here. This is the cofactor. The role of cofactor is number 1 to transfer. Let's suppose here is a substrate. I can write here. This is our substrate S. This is our substrate S. Maybe an atom like hydrogen ion or electron, or a group of atom, like acetyl group CH3C double bond O, acetyl group, or just methyl group CH3, or aldehyde group CH3C double bond OH, or ketone group. These are the groups that can be transferred from one point to another point, within the substrate, or from substrate to the coenzyme, and from the coenzyme to the substrate. This is called transfer. So you can say that hydrogen can be transferred from the cofactor to the substrate, or from the substrate to the cofactor. Or this aldehyde group, the acetyl group can be transferred from one place, between the substrate and the coenzyme. Similarly, an aldehyde or ketone group can also be transferred. So this is the basic role of coenzyme, that the coenzyme are involved in the transfer of atoms or group of atoms between the coenzyme and the substrate. This is the first rule. When we proceed to the chemical reactions, then their mechanism, let's suppose we are in glycolysis and Krebs cycle, when we are describing one specific step of a chemical reaction. In that step, we say the enzyme involved is converting the structure A into B. The mechanism, if you want to understand the mechanism, you must bring the coenzyme. You must bring the role of the coenzyme. And in upcoming video lectures in the metabolism part, we will definitely discuss this as well. Okay, this was the first. Transfer of atoms or groups, hydrogen, amino group, and this may be aldehyde group, C double bond O, maybe ketone group or acetyl group. The second function is the recognition of the substrate. Recognition means this is our enzyme and this is the cofactor. Here, let's suppose we have substrate glucose. Suppose there is glucose substrate. The glucose is the substrate for this enzyme, but this enzyme cannot attach glucose with it because enzyme cannot recognize that this is my substrate. Enzyme don't have eyes to see that this is my substrate. So who will help? The cofactor which is attached, it will help the enzyme to recognize its substrate by attaching the substrate with itself. Let's suppose this is coenzyme, and the coenzyme will attach glucose with itself. Once the glucose is attached to the coenzyme, then enzyme can recognize that this is my substrate. So then glucose can be catalyzed. Here you can see coenzyme is an example is UDP. UDP is what? UDP is a coenzyme, uridine diphosphate. Uridine diphosphate is a coenzyme. I have just mentioned shortcut name of this. No need to write the structure of the UDP, but UDP is just a coenzyme. And when it attaches glucose, it becomes UDP glucose. UDP glucose, coenzyme and substrate, these are attached, and now this is then recognizable for the enzyme. Another example is coenzyme A attaches with acetyl group to become acetyl-CoA. So this is again for the recognition. Third, some coenzymes are derivatives of vitamins. We have not yet discussed the chapter vitamins in the last 2 years, but in this year, inshallah, that we will start vitamins. Here, not all vitamins, but some vitamins, especially the B complex. You know, there are fat soluble vitamins A, D, E, and K. Water soluble vitamins. In water soluble vitamins, you have vitamin B complex, vitamin C. So vitamin B complex contain a lot of vitamins. Some of these vitamins become the part of they just become coenzyme. Like, I would like to tell you some example. And this means that vitamins are so much important. Vitamins. For example, thiamine pyrophosphate, TPP. Thiamine pyrophosphate, it is a coenzyme. And this coenzyme came from thiamine. Thiamine is what? Thiamine is a vitamin. Thiamine is a vitamin. From the vitamin thiamine, we get coenzyme. And what will be the role of this coenzyme? It will transfer aldehyde. Yes, I already told you that in the transfer of aldehyde. So this coenzyme helps in the transfer of the aldehyde group. The example of that enzyme which contain TPP as a coenzyme. So this is transketolase enzyme. The name of the enzyme is transketolase. Its coenzyme is TPP. Then we have another example is flavin adenine dinucleotide. Flavin adenine dinucleotide that we can write in shortcut as FAD. Flavin adenine dinucleotide. Of course, it is the combination of nucleotide, nucleic acid, from nucleic acid, and the vitamin flavin. So flavin adenine dinucleotide. This is another coenzyme and very well known coenzyme, FAD. You might have heard FAD, NAD. So FAD, complete name is flavin adenine dinucleotide, is itself a coenzyme. At least our student can now recognize, can now feel that FAD is a molecule and it is a coenzyme. From which vitamin? From riboflavin. Riboflavin is vitamin B complex vitamin. Riboflavin. From this we derive FAD. And what will be the role of this FAD? FAD helps in the transfer of hydrogen or electron. As I already mentioned, the example of that enzyme is amino acid oxidase. Amino acid oxidase this is an example. Then we have another example is coenzyme A. Coenzyme A. This is another very well known coenzyme. You might see this coenzyme in lot of chemical reactions in metabolism part. From which vitamin? From pantothenic acid. Pantothenic acid is another vitamin B complex vitamin. Like riboflavin thiamine, it is another vitamin. So from this vitamin, we get coenzyme A. And what is the role of this? To transfer acyl group. What is acyl group? Acyl group is actually CH3 C double bond O. This is called acyl group. So in the transfer of acyl group, the example of that enzyme is thiokinase enzyme. So this table is very much important for you. If the question comes or write something on cofactor of coenzyme, you will give the example on this table. Although there are a lot of many examples available, but I think the 3 examples are enough for today's lecture. Now, non-vitamins coenzyme. Some coenzymes are not from vitamins. So we must know that all vitamins are not involved as a coenzyme. Some coenzymes are not vitamins at all, simply. That is adenosine triphosphate ATP. You might have heard this word. ATP is a source of energy. ATP is not a vitamin, but still it can act as a coenzyme. UDP, uridine diphosphate, I just mentioned its name here, UDP. Uridine diphosphate is another coenzyme. These are not from vitamins. And some of the coenzymes are from the nucleotides. You know, nucleotides are the smaller units of DNA, RNA, nucleic acid. So some of the nucleotide, they become coenzyme, like FAD. Now here is a very interesting thing here. The interesting thing is that the FAD is from both sides. Number one, it contains flavin adenine dinucleotide. This A is for adenine. Adenine is a nitrogenous base. Okay. Adenine is the part of the DNA. Adenine is a nucleotide. Adenine is a double ring nitrogenous base. So from this A point of view, from the adenine point of view, we will say that Coenzymes are from the nucleotide as well. But this flavin, which you can see here, flavin is again from the vitamin side. So in this coenzyme, F.AD coenzyme, we can see the vitamin part is also there, the nucleotide part is also there. Flavin adenine dinucleotide, it is still a coenzyme. Another one is NAD, nicotine amide adenine dinucleotide. Here, N, nicotinic acid. This is from vitamin. N is again from vitamin part. A is from the nucleotide part. Okay, NAD. So nucleotide, here adenine nucleotide is there, adenine nucleotide is there, adenine nucleotide is there. Therefore, we call it nucleotide coenzyme. At least our students should know that coenzyme can also be from nucleotide as well. So ATP. Now, this is a really very important point. Believe me, for the first time I heard this word from a book, from the book of Satyanarayan, that majority of the coenzyme are non-protein. But there is one example of coenzyme that is protein coenzyme. Which one is the enzyme ribonucleotide reductase uses thioredoxin. Thioredoxin is what? Is a coenzyme, but it is a protein. So it is an exceptional case. You can say that not 100% coenzymes are non-protein, but there is one enzyme, there is one coenzyme which is itself protein, that is thioredoxin. Thioredoxin is a proteinous coenzyme. And what is the name of the enzyme which uses this thioredoxin? It is ribonucleotide reductase. Ribonucleotide reductase is the name of that enzyme. You can just memorize it. Next point is coenzyme do not decide enzyme specificity. Last point. Enzyme specificity means one enzyme can perform only one reaction. This is called enzyme specificity. In order to get this idea, I have mentioned two examples here. This is enzyme 1, enzyme 2. This enzyme is LDH, lactate dehydrogenase. Means this enzyme acts on the lactate. So when it acts on the lactate, this reaction will be something else, different reaction. And another enzyme is alcohol dehydrogenase. This enzyme will act on alcohol. Now these two reactions are not same. These two are different reactions. Now we can see here this enzyme LDH use NAD as a coenzyme, and this also use NAD as a coenzyme. Both these two enzymes use NAD. It means that the coenzyme is not responsible for the specificity. If the coenzyme was responsible for the specificity, then NAD, which is a coenzyme here, and NAD, which is also a coenzyme here, these two enzymes would not act on two different substrates. These two enzymes would act on either on lactate or alcohol, but not on both. Now this enzyme is acting on lactate and this enzyme is acting alcohol. Both use coenzyme. The concluding statement will be that the specificity depends upon the apoenzyme, means the protein part, not the coenzyme. Coenzyme usually do not decide specificity. Okay, this was the last point. These were the points and overall points about coenzyme and cofactors. I hope you got this video lecture from about coenzyme and cofactors. Inshallah, in the next week we will march towards another most important topic from the same chapter. Till then, bye bye, take care.